Okay, thank you very much. Thank you for the invitation to this great conference. So today I'm going to talk about coevolution. So I have been working on coevolutionary theory in the past, and so I thought it was about time to do some experiments about coevolution. So I am going to present some work we've been doing on coevolution between bacteria and their phage. So this is the first time I present this work, so any comment or feedback would be very welcome. So in the lab, we use Streptococcus thermophilus, which carries a CRISPR immunity against lytic phages. So I won't have time to detail all the paths that lead to CRISPR immunity, but I just want to highlight that CRISPR is an adaptive immune system that allows bacteria to capture a fragment of the phage genome and to put it in a CRISPR array locus like that. And when the spacer is integrated into the CRISPR locus, the bacteria become immune to the infection to the phage. And what I want to highlight is that when you expose a susceptible bacteria to a single phage, a phage genome, you can generate a lot of diversity, a lot of different types of resistant bacteria. Unlike when you lose a receptor on the surface, which allows also to resist the phage, here you can have different types of resistant bacteria. Different types, because each of these different types can capture different spacers in its CRISPR array. And indeed, for example, in the phage that we are using, we know that there are 700 sites that can be used as a target for the CRISPR immunity. So what we know also is that the phage can also escape immunity by just mutating the target sites, which allows to escape immunity by CRISPR. So it's a very nice system to follow both host evolution and phage coevolution by this mechanism. So a bit more about CRISPR specificity. So in a way, it's a classical multi-locus gene-for-gene system. So if there is only one resistant bacteria like that, so the phage needs to acquire one escape mutation to infect both the susceptible and the resistant bacteria. But as I said, there are many different types of resistant bacteria that can emerge after exposition to this phage. And in fact, acquiring this black mutation doesn't allow the phage to escape the other immunity. So the phage needs to acquire additional mutations to be able to infect the whole bacterial population. So there is an intrinsic asymmetry here between the ability of CRISPR to generate a lot of diversity and the necessity for the phage to accumulate mutation to evade defense. So that's something that has been studied by several groups. So that's something that has been demonstrated that indeed, CRISPR diversity is a way to defend the whole bacterial population against phage evolution. So something that we've been studying in Montpellier, so during the PhD of Hélène Chabat, who is currently in Zurich, we've been also studying the speed at which the phage are able to escape CRISPR immunity. So something that Hélène found is that there is a huge variability in the phage mutation rate along the phage genome. So here what this figure plots is the rate of mutation that was measured using Luria-Delbrück experiments when she tried to follow the acquisition of escape mutation against different types of CRISPR immunity. So for example, in this red bacteria, the protospacer was located in this region of the phage genome, and the rate of mutation was very low. And on the other hand, in this bacteria, where the spacer was located in another region of the genome, the mutation rate was very large. So a naive expectation from this would be that the phage are going to be able to accumulate very fast escape mutation against those bacteria, and it will take a bit longer to escape those bacteria. So I will come back to these predictions a bit later. What we were really interested in looking at is what happens to this diversity of bacteria. When you mix all these bacteria together, how does it change through time? So we did this experiment where we mixed 17 different types of bacteria the 16 resistant bacteria that I just described, plus the ancestral bacteria that was sensitive to the phage. And we did batch cultures over 4 different days. So each day we transferred 1% of the culture. So we did 8 replicates of this first treatment. And in this case, we did not use any phage. So we just wanted to see what would happen to the diversity without phage. Then we did a second treatment where every day we added 10 to the 5 phage during the transfers. So to see what would be the impact of phage infections on this diversity. So I have to add that this phage was only able to infect the naive bacteria, the susceptible bacteria. And in a third treatment, we actually, instead of using the ancestral phage, we used escape mutants. And in fact, we used 16 different genotypes. Each of these genotypes were carrying a single mutation, allowing those phage to escape one and a single one of the different resistant bacteria. So and each time we had 8 replicates, and we wanted to track first the diversity of bacteria. So what we did is actually we tracked the evolution of diversity on the CRISPR locus. Okay, so what we did is a PCR of the leading edge of the CRISPR array, which allowed us to track the diversity because all the spaces are in the CRISPR array. So when you sequence these amplicons, you can actually figure out which bacteria you are dealing with. So in other words, we heard yesterday about using barcoding to follow the evolution of bacteria. So here we are using CRISPR1, the CRISPR array, as a barcoding, which allows to track diversity through time. And on top of that, it's also an evolving barcoding because the CRISPR1 locus can actually acquire new spacers to the phage. And then we sequenced, so the depth was quite large, which allowed us to really track the frequency of the different types of bacteria. We were also interested in phage coevolution, so we also sequenced the phage through time. So in this case, we sequenced the whole genome, so which led to a slightly lower depth of sequencing, but it still allowed us to track the escape mutation through time. So I will present the results now. So I will start with the population dynamics of the results. So again, so there were 3 different treatments, and without phage, so here I'm plotting the density of bacteria through time. Without phage, we saw that, as expected, the bacteria would grow to a carrying capacity around 10 to the 8. And then when we added phage, without diversity, we saw the following. So initially, there was only a little impact of the phage on the density, but then there was a drop in the density, which was actually due to an epidemic in the phage population. And then by day 3, there was the bacteria started to recover and then reach almost the carrying capacity without phage. And the phage also was able to maintain a large density by day 4. In contrast, when we used the diverse phage population, we saw that there was a huge impact on the density by day 1. Indeed, when, as I explained earlier on, so in this treatment, all the bacteria are targeted by at least one phage in this mixture. Okay, so which allows collectively the phage population to create a massive epidemic, which we see here when we plot the phage density, which leads to a drastic reduction of bacterial density. But then again, by day 2 and day 3, the bacteria recover. And again, they reach a carrying capacity by day 4. So quite a lot of variation among replicates. So it's hard to say what's going on without going into the detail of the genetics behind this pattern. So that's what we looked first. So we looked at the host evolution, and we wanted to look at the diversity of the CRISPR immunity on the CRISPR1 locus. So there are different measures of diversity. So here I'm just going to show what we obtain when we look at the effective number of alleles, which is a classical way to measure diversity, and it depends both on the number of genotypes and the frequency of the different genotypes. So when we plot the effective number of alleles across time, so we start with 17, around 17, which is exactly what we did in the experiment, because we mixed 17 different genotypes in equal frequency. So that's what we expected. But what is quite spectacular here is that the diversity is dropping very, very fast. So by day 2, without phages, all the diversity is lost. So there's only one genotype remaining in all the replicate populations. When we add phage, we see that there is also a drastic drop in diversity. In fact, steeper than without phage. But then the population starts to recover and the diversity is maintained. And in fact, it can also increase with time. And again, there's quite a lot of variation among replicates. And when we start by adding a diverse phage population, what we see is that initially the bacterial population diversity is maintained, but by day 2, there is a massive drop in diversity. And by day 3 and 4, the bacteria recovers again. So we see very contrasted patterns and very contrasted effect of the treatments on those patterns. So to dig deeper into what's going on, so we wanted to plot the results in a more meaningful way. So we use Mueller plots. So we saw beautiful Mueller plots yesterday. So we also did that in this experiment. So what I'm going to show here is the bacterial density of the different genotypes that we followed. So, okay, you remember we start by introducing 17 different bacteria. So the gray one is the susceptible bacteria, the GGCC. So it's the ancestral type. And the colored ones are all the 16 different resistant bacteria. And the color code refers to the fitness of those bacteria. And by fitness, what I mean is that what we did here, we just inferred fitness from the change in frequency from day 0 to day 1. So for example, here you see that the red one has the highest fitness because it's the one that increased most in frequency between day 0 and D1. So there are several things to discuss here. First, naively, one could think that because those bacteria are resistant to the phage, you may imagine that there is a cost associated to CRISPR immunity, and that this bacteria could win the competition between the resistant types. And in fact, this bacteria is not doing so badly. I mean, it's actually increasing between 0 and 1, but there are actually other bacteria that are doing better than this type. And in fact, the red one is really doing much better, and it's outcompeting all the strains in the population. What is quite surprising is what happens with the green type, because it's also a very competitive strain that occurs a bit later in the competition. So we're not quite sure what's going on. So we asked our collaborators in Quebec to sequence those 17 different genotypes. And so they found several mutations, and I'm just going to highlight what happens in these two winners in the competition. So in the red type, there, indeed, our collaborator Sylvain in Quebec was able to spot 9 unique non-synonymous mutations. So maybe some of those mutations are beneficial for the bacteria, and that's what is driving this higher competitive ability of the red strain. It's unclear about what's going on because all those unique mutations were synonymous. So maybe there is some pre-existing mutation here that is spreading and allowing this green type to be more competitive by the end of the experiment. We don't know. So that was just one population. So what happens in the 7 other replicate population? So I'm showing this here. And I mean, obviously, the very same pattern occurs. So there's slight variation in frequencies, but it's very, very repeatable among replicate. What is interesting is to look at now what happens when you add phage in the population. So again, here I'm going to show the treatment where we add a phage that is only able to infect susceptible bacteria, the gray ones. And indeed, what we see here is that initially the gray bacteria is dropping because it's been hit by the phage. And what I want to highlight also is that we are able also to track the population density. And we see that there is a drop in the density that varies among replicates, but by day 2, there is a drop in the density of the bacteria population. And what is very striking here is that the red type, the very competitive one, tends to win also by day 1, but by day 2, it's outcompeted by other types of bacteria. And by day 3, another thing is happening. So those new colors, the darker colors, indicate that there is new spaces that are incorporated in the CRISPR locus. So there is a new resistant types that are emerging, and there are. And what I want to stress here is that those new types are actually mostly emerging in the red and green types that were already identified as very competitive. So, and there is variation among replicates, and I want to show also what happens with diversity. So here, with diverse population, the initial drop in density is happening by day 1. And because all these bacteria can find a phage that is actually infectious in the population initially. And what is striking here is by day 2, you see there's a lot of variation among replicates. And again, there's a lot of secondary spaces that are acquired in the CRISPR locus. So one way to measure this variation among replicates is to look at FST, which is a measure of differentiation among replicates. So I'm going to show you this here in this figure. So this is the FST among replicates across time. Without phage, there's almost no FST building up. With phage, with no diversity, there is some FST building up slowly. And when there is a lot of phage diversity, that's what we saw in the previous Muller plots, the FST is actually building up by day 2. There's a massive amount of differentiation. And what's going on here is that the phage has a drastic impact on the bacteria. So the bacterial population is dropping to a very low level. And by day 2, there is acquisition of new spacers. And each replicate is acquiring new spacers. So that's why there is a massive amount of differentiation here. So of course, here, what is driving this differentiation is phage. So in coevolution, classically, coevolution is driving negative frequency dependence. So we are also able to look at that in this experiment. So here I'm plotting the change in genotype frequency as a function of the genotype frequency at a given point in time. So without phage, we don't see any sign of negative frequency dependence. But in the 2 other treatments with phage, we see a strong pattern where there is a massive impact of the phage on the negative frequency dependent selection. So I want to speed up and talk about phage adaptation next. So what I find very interesting with CRISPR is that we know precisely the link between the genotypes and the phenotype in this system. So it's possible, given that we have the genotypes of all the phage in the population after sequencing, to actually measure mean fitness of the phage. And so we talked about HI, which is the frequency of different genotypes of bacteria. And now we can also infer the frequency PI of phage that are able to escape those particular genotype I. Okay. And with that, we can measure the mean pathogen fitness. So there is a twist here is that if there is only a single spacer, it's easy to calculate the frequency of escape mutation because it's the frequency of the escape mutant in the population. When there are 2 spacers, you have to figure out what is the frequency of the double escape mutant in the phage. And because we have only short reads, we have to use an assumption about how to calculate the frequency of double escape mutant. And we have to assume in this experiment, at least in the figures I'm going to show, we assume that there is no linkage between those mutations, which allow to figure out what are the genotype frequencies, the double escape mutant. So I'm going to show, okay, what is the mean phage fitness in those experiments. And what is very powerful here is that because we have genotype frequencies across time in both the host and the parasite, we can measure a lot of mean fitness. We can do a lot of cross-infection experiments that take a lot of time experimentally. But here we can just compute the mean fitness by just calculating this product here. And so here I'm plotting the mean fitness of the phage population when we vary the sampling time of the phage and the sampling time of the host. So if along the diagonal here, you have the contemporaneous match between the time of the phage and the time of the host. So here you have the phage population that is adapting through time. And here in this part of the matrix, you have phage from the future. And you see that they are well adapted to the bacteria from the past because they are below this diagonal. And in this upper part of the matrix, those are bacteria from the future that are fully resistant to phage from the past. So another way to look at that is to plot those kind of graphic where you can plot phage adaptation as a function of the lag between the timing of the sampling of the host and the parasite. And you get these typical patterns, which are actually expected from the theory, where phage from the future are able to infect the bacteria, but phage from the past are doing very bad. So we can do that for the other treatment as well, and we get similar pattern here. And just one thing I want to highlight here is that with those patterns, you tend to average out what's going on for different lags. But what we see here, for example, is that the phage are actually adapting to the bacteria from time 2. And I think it's very interesting that we are able to do that in this experiment. And maybe this is something that can happen in other coevolution system as well. So we can look also at local adaptation. And so in the first treatment, we see no local adaptation. But when there is diversity, we see a strong pattern where the phage is locally adapted. And what is interesting here is that there is a lot of local adaptation here precisely because there's a lot of differentiation among host population that is driven by genetic drift in the bacteria population. So there is a link between genetic drift and local adaptation, which was also something expected from the theory. So I want to discuss briefly the initial hypothesis about the rate of mutation. So again, I show you there was variation in mutation rate. So we were curious to see what would happen if we track escape mutation to these different resistant bacteria through time. And so we were expecting to see a lot of escape mutation here, in this part of the graph, especially in this treatment with no diversity. And what we found, actually, so here I'm barring the time, in fact, is that early on we see a lot of escape mutation in those types here that were supposed to be very difficult to escape. So it's kind of counterintuitive regarding our initial prediction. And to make sense of what's going on, you have to return to the competition assay that we did without phage. And then it makes sense because you see that those phage are actually adapting to the most competitive types in the system. So the winners are driving phage coevolution in this system. So just a take-home message. So I want to recall what's the classical view of phage and CRISPR immunity is that phage are going to drive a lot of diversity. This diversity is going to hamper the evolution of the phage and potentially lead to phage extinction. And what we found is that together with this acquisition of new mutation, of new spacers, the bacteria are going to acquire new mutations. So, and these new mutations may allow some variation in the competitive ability of the bacteria, which will lead to a lower diversity. And this lower diversity then will allow phage to escape and maybe coevolution to go on for longer. So with that, I will end, and I want to thank my collaborators so we've been working with people in Aarhus, with Thomas Bataillon, and we have a long-term collaboration with Sylvain in Quebec. And I want to highlight the help of Martin Guillemet, who did all these great figures. Thank you.
Okay, thank you very much, Sylvain, for this very interesting talk. So